Thank you. Hello, my name is Erin Zwack, and today with my colleagues Samantha Simpson and Sabria Rosemond, we're going to talk about our collaborative work with Missouri Western to build an E. coli computer to solve the burnt pancake problem. To engineer this computer, we have already designed many different parts, which we have been able to test and include in different devices, as you can see here. So what is a biological pancake? A biological pancake is a sequence of DNA that is flanked by Hicks sites. When Hin recombinase is added, it's able to take the DNA that's between these Hicks sites and flip it around. When we originally started our project, for design reasons, we hoped to make one of our pancakes a promoter RBS pancake, but upon testing, we found that the ribosome is unable to read through the Hicks site, and thus we had to combine the ribosomal binding site into the coding section of the pancake, as you can see in this construct here. We hope to use these constructs to solve the mathematical burnt pancake problem. As you can see in this picture, we have a stack of 4 pancakes. These stacks can be represented as signed permutations, with the number being equivalent to the size of the pancake, and the sign, positive or negative, being equivalent to the orientation. So if you have a negative sign like here, the burnt side of the pancake is facing up. To the right, you see red arrows. These arrows represent where the spatula can be inserted into the stacks. Using the formula n+1 choose 2, we were able to figure out the different positions that the spatula can go into. We, for modeling purposes, originally assumed that all different positions for the spatula were equally likely to happen, and using MATLAB, we simulated what the results would be. In this graph, you see the results of the simulation for 8 stacks of 2 pancakes. Each line in the graph represents a different stack, and there are multiple stacks in each of these sections. There's actually more than one line, though they kind of fade together. On the side, you see the percent of plasmids that solve the problem, and across the bottom are the number of flips it took. As you can see, you end up with different clusterings of stacks that solve it in about the same manner, and this actually is determined by how many flips away from the end of the problem the original stack was. We, for purposes, have called these families. Continue. You can also notice that after 5 flips, the families become indistinguishable from one another. Therefore, we need to be able to control the flipping of Hin so that we can stop it before it reaches the 5th flip. Continue. Using this, we have created different pancake constructs. The top one shows what would happen with the promoter in the same direction as the RBS, and as you can see, the promoter is the flippable unit, which is designated in the number section by having parentheses around the 1. With these constructs, you expect that the first and third would have TET resistance as the promoter and the coding are facing in the same direction. But at the same time, you would also expect the ones with the coding and the promoter facing in different directions to be TET sensitive. Interestingly enough, when we tested that, that was not the case. This, we determined, was from transcriptional read-through from the plasmid. Although the promoter was not actually causing this to happen, a promoter outside of our device was causing the TET resistance gene to be transcribed. As we couldn't, therefore, use TET resistance to screen for if the problem has been solved, we did a restriction map of the device and vector and found 2 unique NHE sites in our constructs. As you can see, each site will give a different fragment band depending on the orientation of the construct. So we went into the lab and we tested this. And what we found was that when you just had the 1, 2, you had a single band, and when you had negative 1, 2, you also had the single band at a different position. But when we added the HIN gene to our constructs, you see 2 bands, which means flipping has occurred. When we did it with TET, you see the same result, the single band for without HIN, and once HIN has been added, you have double bands. While our HIN cassette did have an inducible promoter, we did not actually have to induce the promoter to get HIN to be produced, which once again shows that read-through is happening with these plasmids.
All right, so Aaron was just talking about how we had some read-through with our plasmid, and so we created a new plasmid, PSB1A7, that had double transcription terminators at the end of the device, so that way you could put the whole entire device, including the biobricks, in between the transcription terminators in this vector, and there would be hopefully no read-through through the plasmid. So we went ahead and we tested this, and we got observed test resistance in PSB1A7, and it worked the way that it was supposed to, so we know that the vector that we created worked. Next slide. So just to go over all of our original design problems, first of all, we had the two double terminator sites within the plasmid that made it unclonable into our old vector, so into PSB1A7, so somehow we had to get rid of the double terminators within the plasmid because of inverse repeats that made it unclonable. Also, the recombinational enhancer at the end caused the HIN protein to work too quickly and actually flip the Higgs sites kind of uncontrollably, so we wanted to have more control over our construct, so we wanted to figure that out as well. So the solution to that was two vectors into the E. coli. The first vector would have 2 pancakes, and that would be put in our PSB1A7 vector. So that way, all of the tetracycline resistance or any sort of antibiotic resistance we got would be from that device. And so PSB1A7 still held our pancake construct. And then we had to use a separate vector that had ARC and HIN in it, and that plasmid does not have the transcription terminators in it, so therefore we could still have the transcription terminators within that device. So those were the 2 plasmids that we used. And then a second problem that we had with our original design was the biological equivalence problem. Both 1, 2, and -2, -1 would look the same when plated on an ampicillin, kanamycin, and tetracycline plate because they'd both be resistant, so they'd both be resistant to all of those antibiotics, so they'd both grow. So an easy way to tell which orientation was which was to add RBS, RFP sites at the beginning of the pancake device, so that way if it was in the -2, -1 orientation, if the promoter was in the -1 orientation, those colonies would be red when they were plated out. So that way now we had 2 unique solutions to the pancake problem, so 2 solutions that would grow on a tetracycline plate. Next slide. That now gave us 8 unique 2 pancake stacks. They're grouped by the families that were in our earlier mathematical modeling graph. The first 2 would be the only tetracycline resistant ones, and that's why they have the blue background. And then the ones with the red would be red because the RBS would be expressed when grown on an ampicillin plate. Next slide. So we built our new pancake design, and we tested that as well. So the first column of this is to see if we could repress PBAD, which is the promoter, within the pancake construct, and we could because when we added ARC to the construct, the 1-2 pancake, which should grow on a tetracycline plate, did not grow, which showed us that the ARC-HIN complex was working. The second column is looking at whether or not we could activate the PLAC-HIN construct. So the HIN vector, we added with IPTG. So we added IPTG, and we showed that HIN and ARC were both producing proteins, so we know that that's working. And the third column is referring to just this, if we could screen for orientation, and if the orientations that we thought should be tetracycline resistant were tetracycline resistant, and they were. So we have all three of those working, all three of those separate areas working, and the next steps are to create a construct that has all of these parts working together. So those are the next steps for our design.
Aaron and Samantha have said a lot of information, but what does all of this mean? There are practical implications and there are also biological implications. The practical implications include the proof of concept for bacterial computers. We created the first bacterial computer, and the usefulness of having a bacterial computer is that you have a multitude of parallel processors. This increases your chances of having at least one of your plasmids solve the problem. Also, you have a range of data storage, and you can store data is stored in the manner of either order and orientation, or a combination of the two. The basic biological research, you have improved transgenes in vivo. This mechanism can act as a switch to turn your gene on and off, so you have a choice of when you want your gene to be expressed. Also has evolutionary insights into homologous chromosomes and how they've changed over time through the process of evolution, as in from the mouse to man. Slide. Before we can get there, what do we need to do? First of all, we need to slow down HIN so we can control the kinetics of the reaction. By slowing down HIN, we can find out how many flips happen per second, and also find if HIN has a size bias. Does it prefer the smaller promoter or the larger coding region? Slide. Also, we need to think about how the plasmid copy number affects the outcome of the cells. We've created a mathematical model that shows the probability of how many cells will remain alive based on the probability of the plasmid solving the problem. In the first row, you have a two-stack construct with 200 plasmids in the cell. Only one needs to solve the problem. There's a 25% chance that at least one of the plasmids will have a solution. But with this, you see all of the cells survive, so you can't tell the difference between the families. In a four-stack construct, you with 200 plasmids and one needed to solve the solution, you have a 0.3% chance of one of the plasmids solving the solution. About half of your cells live, so you're starting to be able to learn a little more. Also, a four-stack construct of a different family with 200 plasmids, or when one has to solve the problem, you have a 10% chance of one of the plasmids solving the problem, and you have about 87% of your cells living. This will allow you to be able to tell the difference between the families and how they're acting. Slide. Also, we need to find an improved insulating vector that will be able to accept the double terminator and also look into making bigger pancake stacks, which can store more information and possibly solve more complex problems. Slide. As Missouri stated, multiple campuses attacking a problem at the same time, you have more ideas, more minds working on something. Somebody sees things from different angles, so it's really helpful. Also, when you're having a problem, you all can, as we had a lot of conference calls and video conferencing, so we were able to figure out how we could troubleshoot or who was going to attack what problem and how, and it made the wet lab more effective. Also, something that we learned was that undergraduate schools who have no graduate programs, small schools with no graduate programs, are able to compete in iGEM. Slide. Math and biology students work really well together. As you can see here, this was Davidson's attempt at making the graph that you saw on their sphere. We were able to prove a new theorem, which was awesome. Also, collaborating with math allowed us to approach our biological wet lab more effectively, because we were able to model what would be the best way to do things. Slide. But above all, we had a flipping good time. We'd like to thank our collaborators at Missouri Western State University and all of our supporters.
If there are any questions?
What was the role of Hampton University?
Oh, I go to Hampton. I'm actually not a Davidson student. So this was actually a trial run to see how students from other universities could come together.
Your immediate results, your intermediate results, were you able to prove that your modified C construct did not flip when unused?
Actually, the HIN complex, we did Western blotting, and there was a small, in the first pancake construct, we saw some flipping without IPTG being added to the HIN complex. So there is a small amount, but it's much greater amounts of protein are present when IPTG is added to the system. So there is a small amount of HIN activation, yes.
So as I understand it, the bacteria don't actually solve the problem. They make guesses at the problem, the solution, and then you kill them if they guess wrong.
Yes, exactly.
Our hope is to be able to control flipping a lot more precisely than we can. So if we're able to know that after 5 minutes it will have had the chance to flip once, we could then stop it, plate it, and then the ones that survived have solved it in one flip, or more likely than not have solved it in one flip, and the ones that have died were unable to solve it in one flip.
How do you propose at stopping it and plating it, or, as you were discussing, determining flipping per second?
Yeah, that's something that I think we're still working on. I don't know if the math modeling is.
I mean, I think you're asking about the specific biological construct.
It would basically be a control of HIN and the HIN protein, and most likely finding a way to stop its production, which would be probably controlling the promoter in front of the coding region, and also removing HIN from or enabling the HIN protein that's already in the cell from inverting more DNA.
Okay. Well, yeah, it seems like what you might want to do is couple it to something like initiation of cell division, so you know it happens only rarely.
Yeah, that would be a good idea.
Thank you.
I was thinking also of attenuating that protein in some way if you're able to mutate it or target the sequence. If you could make it a less efficient process, perhaps you'd have more control.
Thank you.
Also a very good idea. Yes, we like these ideas.
I know Lance is a good cook. What if he makes two pancakes exactly the same size? Mathematically, what happens?
Do you mean, I mean, one thing we looked at with the modeling was if there would be a bias, since those parts are different lengths a lot of times, there would be a bias in the flipping. Now, I don't know if you're asking that question or if you're asking if we had two pancakes that were composed of the same DNA sequence.
I'm asking mathematically. If you were flipping and you were making the assumption that your stacks were different size, different diameters. So now we have two that are exactly the same. Now what happens?
As far as trying to solve the pancake problem?
Right.
I mean, the question is, would it be the same solution if those two pancakes, you know, if you call them both number 2 or 1, 2, and 2 prime, if 2 before 2 prime is the same as 2 prime and then 2, and I mean, I don't know. I think that's
Are you using size as a detection method? Is that why you're using it?
Yes.
Yeah, I think you're only using size as a detection method because the fluorescence wasn't working? Or
Within the original pancake problem, that there's a stack of pancake of all different sizes. And it's just, in that problem, they never really say that there's two pancakes that can be the same size. It seems like it's just assumed that all of the pancakes are different sizes.
Any other questions?
No further questions? Thank you, speakers.